odd day classes. The quiz is on the 18th. If you're watching the video, I've got to fix this for even day classes since I was absent, so actually odd is ahead of even, so yours will be shifted down. Your lab will be due on the 22nd, and then Unit 2 test is the 26th, and then before you know it, it'll be cool weather all the time, and it'll be October, so it'll be great. Any questions about schedules or anything? Again, odd is exactly the same up here. Nothing has changed. Cool. All right. So for us, we are picking up where we left off. So if you have your notes with you, get those out. The notes look like this. So they look like that. All right. So last time we were in class, we were kind of up here in this area on our notes. We didn't get super far. We spent most of our time talking about the test and stuff like that. So remind me again really quick, how does this picture relate to a phospholipid? Huh? Yeah, Joe. Yeah, this is the base structure for a phospholipid, and phospholipids have two tails. So I basically just cut off one of the triglyceride fatty acid tails, and I replace it with a phosphate. I spin it around, and now I've got a phospholipid. So phospholipids, a lot of times teachers draw them kind of like that. Those are the two tails. Usually one tail is straight, it's saturated, and usually one tail has some bends in it. So that's a phospholipid. What was the round part on top representing? Yeah, the phosphate. So that was a phosphate, PO4. And then what's significant about the phosphate? Yeah, it's polar. It's got some negative charges on it. What about the tails? Are those polar or nonpolar? Nonpolar. All right, cool. So the phosphate head was polar. The lipid tails are nonpolar. So those are nonpolar lipid tails. And then if I had these phospholipids, would they be more like liquidy or more solid? Yeah, more liquidy because they're unsaturated. All right, so then somewhere in here, did I draw a picture of a beaker? Okay. So we had a beaker up here. I poured a layer of phospholipids, and they all ended up kind of face down in the water. And then if I had another layer of phospholipids, we saw that they went across the top like that. And again, I'm being lazy. Every other one of these tails is typically going to be unsaturated. Let me take a roll really quick. I think everybody from KCal made it. Except Ray. Okay, cool. So I've got this phospholipid bilayer, and what happens if I dunk it underwater? Okay. Yeah, it makes like a cell. Did we show that in class? Kind of loops back on itself. So picture this as like here's one layer of phospholipids, maybe the bottom one, and here's the top one. I don't have black and purple. I've got yellow and purple. So here it is. Picture like a peanut butter sandwich and pretend that the peanut butter can't touch the air. So from above, that's all bread, right? All the peanut butter is like in between if I like open up the sandwich. But on the very, very edge, wouldn't like right where the edge is, bless you, wouldn't there be like a little thin line of peanut butter in the middle? Wouldn't it be like touching the air? So what form would this have to take to make sure that like only bread touches the air? A sphere. So this is a square. It won't fold up into a perfect sphere, but like basically if I can get this end and this end to touch, then it's bread all the way around the outside, bread all the way on the inside, like it's a sphere. Does that sandwich analogy make any sense at all, or is it just stupid? And you're like, well, it's both. It makes sense, but it's also stupid. So I now have a cell like this, and then one of the last things that we mentioned was like, how thick is this phospholipid bilayer? Yeah, thinner than paper. In fact, we think it's about 1/9,000th the thickness of a sheet of paper. So 9,000 of them stacked on top of each other is the thickness of a sheet of paper. And so we left off with me just being in amazement that this is what holds us together. So the purple phospholipids on top, they're not bonded to each other. The phospholipids on bottom, they're not bonded to each other. So imagine this is a phospholipid. That would represent the head. The rest of the pin would represent the tails. It kind of looks like this. All the heads are facing up, all these heads are facing down, and they're just like sliding past each other because they're not bonded to each other. So those purple guys are not bonded to their neighbors. These on the bottom part are not bonded to their neighbors. The black and the purple are not bonded to each other. And they're liquidy, and they just like slide past each other. How thick was this barrier again? 1/9,000th the thickness of a sheet of paper. Was it solid or liquid? Liquid. Are the individual components even bonded to each other? No, and yet that's what holds me together. So I can play football, I can box, I can fall down the stairs. The last one's on accident. And so I can do that, and like I somehow managed to not die. Okay. Let's say that my phospholipid bilayer, kind of like we were saying last time, was like phospholipid bilayer, and it's floating on the surface of water. And then I go in and I like karate chop the water, and it folds up. If this is now like underwater, then everything around it is water. Right? If I like knock this below the water level, what's in the middle of it? Water. If my hand is floating on the surface of water, then right here's the surface, and I go like this, I dunk it underwater, and I go like that, then now my hand is surrounded by water, of course, but I've also got a handful of water in the middle, and that's the beginnings of a cell. Why don't these purple phospholipids or these black phospholipids like leave? Like why doesn't one of these purple ones kind of like scooch out, or one of these scooch out? Yeah, those lipid parts in the middle don't want to touch water. So as soon as this guy would like start to head out, he gets sort of like pushed back in by water. And that's all that holds your cells together. Purples aren't bonded to each other. Across the aisle, purple and black is not bonded to each other. The only thing that's keeping your cells from falling apart is the fact that that middle lipid layer doesn't want to touch water, and you are like 2/3 water. That's all that's holding you together. What's holding my cells together is 1/9,000th the thickness of a sheet of paper. It's made of liquid. The components are not bonded to each other. And the only reason I'm still here is because they're surrounded by water, and the middle parts don't want to touch water. That seems weird to me. You might be like, that's kind of how I thought it was. But to me, that's kind of shocking. Imagine the bricks of Timber Creek. So let's say the brick walls at Timber Creek are like this wide. And there's no mortar or nothing. All the bricks are like, they flow, they move, almost like if you're in a bounce house and you bump into the side and the whole wall goes like this. But imagine the middle of the bricks just don't want to touch air. And that's what holds Timber Creek together. Anytime somebody bumps into the wall, the bricks don't slide out because the middle parts don't want to touch air. So the wall just kind of does this. It's good that they're fluid because now I don't shatter. If they were solid, I'd be shattering myself right now. But instead, the walls just kind of go like that. And that's what holds me together. I'll show you this picture a few times. I might have shown you this last time. This will probably be the last of review stuff if I did show you. But this is zoomed in on a single cell. Did I show you guys this last time? Okay, so now we're on to new stuff. This is a single cell. For reasons that we'll discuss later, the default shape for all cells is a perfect sphere unless we force them to be something else. So if I don't do anything special to a cell, it'll be a sphere. You might be like, hey, Mr. Cotton, I thought nerve cells were like long and skinny and red blood cells were kind of like pancaked in. That's true. We can do something to make them not spheres, but otherwise they're perfect spheres. Okay, so this is zoomed in on one cell. That right there is the phospholipid bilayer. If you're in the very back, you might not be able to see it very well, but they're kind of showing you that like that little part right there is phospholipid bilayer. Okay, so we're so zoomed in on this cell that's a perfect sphere. Like that's the arc. So if this is a perfect sphere, then isn't the rest of this circle probably like, doesn't it probably go down like to the floor? Like isn't this like a 5 or a 6 foot diameter circle? If that's the arc, if I like finished off the circle. So we're so zoomed in on a cell that we're zoomed in to where an individual cell is like, you know, I'm almost 6 feet tall. So an individual cell is like 6 feet in diameter. How zoomed in are we? A cubic millimeter of human tissue is supposed to contain 1 billion cells. So in this cubic millimeter, not centimeter, but millimeter, the smallest measurement on any standard ruler you have, a cubic millimeter is supposed to be 1 billion cells. We zoom in on one of those and then zoom in so much to where one of those is 6 feet in diameter, and then that's how big the cell membrane is. It's liquid. Those parts aren't bonded to each other, and that's all that's holding me together is that little part in the middle that looks kind of clear, kind of like oil floating on water. That little clear part doesn't want to touch water. And that is life. Just a coating of oil on your cells. DNA doesn't have to tell them to be in there. DNA has to manufacture phospholipids, except for the ones that I eat. But cells will just automatically make themselves. They just go into bilayers, and they'll make little spheres as long as they're underwater. Kind of crazy. One of the reasons that I like biology is stuff like this just sounds super fake. The DNA sounds super fake. Why is your heart built the way it is? Well, you have this like ladder, and there's A's, T's, G's, and C's, and then you build a heart. Or A's, T's, G's, and C's are wrong, so you have this heart defect. And you're like, well, that sounds like bull crap. And then you're like, well, let me tell you about cell membranes. You're just made out of oil. You know, if you were like designing a robot and you said, I'm going to take 80 to 100 trillion cells to build this robot, and every single cell will be held together by not titanium, but like a layer of oil, I would be like, that's stupid, you're fired. That's the dumbest idea I've ever heard. But that's life. So we're just a layer of oil. Super weird. At this next picture, they have lateral movement. That's what I was showing you earlier. I've got phospholipids are kind of set up like this, and they can move side to side all day long. Why is a flip-flop super rare? Why is that really common? Them sliding back and forth left or right. But why is a flip-flop? Why is a purple and a black phospholipid switching spots super rare? Yeah, the tails have to touch water. Now, you're like, hey, what do they mean by frequent and what do they mean by rare? I'll tell you. So here's what they did. This is how you realize how fluid your membrane is. This is a kind of an analogy version, but this is essentially what they did. They shoot some type of beam at it, laser, light, whatever. And what they did was they just sort of like shot a light at this certain spot on a cell. And anytime a phospholipid moved in or out of that spot, they could see it. So imagine this. Pretend that this chair, I've got a chair up here. So pretend that this chair has like a mirror on it. And you're above it, like shining like a laser light down on it. So anytime you shined a light on the mirror, it would bounce back up and like hit you in the face, right? Okay. What happens if I sit down on the chair on the mirror? Would the light bounce back anymore? No. So essentially that's what they did. They shot a beam of some type of energy. Again, I'm just going to say laser. I don't know that it was a laser. And they would just look to see how many times things like shuffled in and out of that spot. So right here, the red light would go away. Once I get up, the red light would reappear. Somebody sits here, the red light goes away, it would reappear, right? If you're from above, that's what would happen. So they found that on average, phospholipids move in and out of one spot about 10 million times per second. So let me demonstrate that for you. That's like 1, 2, like musical chairs. Somebody else comes up, 3. Only it's 10 million times per second. And so they call that frequent. I mean, you start getting down to like, if you're just going to put dots on a paper and try to go as fast as you can, just like, oh, that's fast. Well, yeah, you're doing probably like 4 dots a second. This is 10 million dots per second. So now imagine every time that I'm hitting it, it's 250,000 times or something. Rare. Often does rare happen? About once a month. So frequent, 10 million times per second. Rare, a dozen times this year. We've been in school for about a month. So pick a cell in my body, and this has likely happened once since we've been in school. And they go, we'll call that rare. Those exact numbers I'll never have to know for an AP Bio test or whatever. I'm just trying to get you to see, when we say the phospholipid bilayer is fluid, we mean it's fluid. In musical chair terms, we're doing musical chairs at 10 million times per second. All right, cool. Why is it that these phospholipids are more fluid than these? Like, what's causing? Pretend that they didn't tell me these were more fluid. Pretend that there were no labels up here. Why is it logical that that's a more fluid phospholipid bilayer than that one? If I didn't have the labels to just tell me. Yeah, it's more unsaturated. So is it possible that my liver cells need to be a different consistency from my lung cells, from my brain cells, from my red blood cells, from kidney cells, whatever? Sure. And so they are. So sometimes this particular cell needs to be more solid. And so I'll just have a small number of unsaturated tails. Sometimes I'll need a cell membrane to be a lot more liquidy. So I can just switch out the phospholipids and make the tails more unsaturated. So your body is actually tweaking this all the time. Sort of interesting. Now, the outside layer and the inside layer, I made these 2 different colors, not just so you could tell like there's 2 layers, but because usually on most cells, the majority of the time, the outer layer of phospholipids and the inner layer of phospholipids are different. This is super clever as to why. So whether it's a liver cell, a lung cell, a brain cell, whatever, usually whatever kind of phospholipids are on this outer edge, the inside is a little bit different. And you might be like, what do you mean? If they're all phospholipids, aren't they all identical? No. So we call them all phospholipids because they all do have the lipid tails. That glycerol molecule, they do have a phosphate, but then they can have other crap attached to them. So nerve cells have phospholipids with some sort of crap attached to it. Liver cells have phospholipids with other types of crap attached to it. All of them are polar because they all have that phosphate head, but they can have other things attached to it. Okay, here's why this is interesting. There's an enzyme. If it's an enzyme, what 3 letters does it end in? If I've got an enzyme, what 3 letters do enzymes usually end in? ACE, A-S-E, yeah. So not even joking, there's an enzyme called flip-ACE and an enzyme called flop-ACE. And flip-ACE and flop-ACE can switch these phospholipids. So they can take one of the internal ones and put it on the outside, or vice versa. I don't remember which one goes which way. Flip maybe takes the inner ones and puts them on the outside. Flop-ACE does the opposite. And you're like, why would you do that? To tell your immune system that you are sick. So imagine this. Imagine all of y'all are Mr. Cotton cells of some type. Liver, lung, brain, heart, whatever. And I'm a white blood cell, an immune cell. The way that I decide who to leave alone and who to attack is I check all the phospholipid layers on you and I check all the proteins that are on you. So I basically go around and I check everyone externally. So I'm just sort of like checking, and I'm like, all right, I bump into this student, and I'm checking all the phospholipids and proteins and sugars and stuff that are on the outside, and I'm like, that's a Mr. Cotton liver cell. Leave it alone. I bump into this student, this cell. That's a Mr. Cotton brain cell or whatever. I bump into this. Cool, I leave it alone. I bump into this, and I don't recognize it. So I'm an immune cell, a white blood cell, so what do I do if I don't recognize something? I kill it. So if this happens to be E. coli, I'll be like, I don't know what that is, and that's how I know to attack E. coli. Let's say there's a brand new bacteria that my immune system has never seen before. I should still be able to fight it because I just know it's not Mr. Cotton. The way that your immune system works is it has a list, so to speak, of all the proteins. It recognizes all the proteins on all of your cells. And if it bumps into anything else that it doesn't recognize, it attacks. So let's say there's a brand new strain of E. coli that's going to come out in 2030. How does my immune system know to attack it? Because it's not Mr. Cotton. It's kind of like group sports. If you and I are on the field and I turn around and you're wearing purple and gold, you're my friend. If you're not purple and gold, I punch you in the face. Right? I don't have to identify as this person from Central, Keller, Carroll, whatever. It's just like they're not wearing Timber Creek colors, so I'm punching them in the face. If I've never met the school before, it doesn't matter. They're not me, so I attack them. Some of you guys, your body confuses food for bacteria. So me, I can eat peanut butter. But for some of you guys, your immune system thinks that peanut butter is a bad guy. So if you eat peanut butter, you have this like major allergic reaction to it, your lips swell up and everything else. That's your body's immune response. Me, I have seasonal allergies. So I wake up, probably blow my nose 6 or 7 times every morning, 10 months out of the year. Ironically, one of the things I'm most allergic to is cottonwood tree pollen. So I'm allergic to myself, kind of. So whenever I breathe in this pollen, your body is like, that's just pollen. It's plant sperm. It's disgusting and I feel violated, but like, it can't harm me. My body is like, oh my God, we're being attacked. We better capture all that stuff in the air in like snot. And then I take another breath and I breathe in more. It's like, we're being attacked again, make more snot. And then I'm like, it's like, we're being attacked again, make more snot. And it's like, it's getting in his eyes, make his eyes all watery. And it's like, it's in his throat, like cough it up. And so you start hacking and everything else. You're having an allergic reaction because your body is overreacting to something that shouldn't matter. What I want my immune system to do is basically recognize all the Mr. Cotton cells and leave them alone. Recognize food as food. And then anything else that's an invader, then I want my immune system to attack. But if I have an allergy problem, then my body is just confused. My immune system is freaking out over something that is just peanut butter or pollen of some kind. Now, here's where it gets tricky. I've got a liver cell, so it looks like this. I've got a liver cell that's cancerous, so it looks like this. Cancer is hard to cure. Cancer is hard to fight because it's my own cells. So imagine there's me, and then there's a clone of me, but this one is misbehaving. You can't tell by looking at me. So they come up against this liver cell, and they're like, that's a liver cell. And it's a healthy liver cell. My immune system bumps into this, and they're like, that's a liver cell. But it's a growing out of control cancer liver cell. That's why cancer is so hard to fix. E. coli looks different. So I can identify that. That's a prokaryotic cell. It's got cell walls. That's a bad guy. We don't have cell walls. But this is one of my own cells. So one of the ways that my cells try to help is they narc on themselves. So flipase and flopase will do this. Let's say that I'm a liver cell and I'm cancerous. If the cell figures this out, in Unit 4, we talk more about like how does this work, how does the cell like know stuff, how does the cell figure this stuff out. So we do talk about this more in Unit 4, but if my cell figures this out, flipase and flopase will switch out some phospholipids. And now, when one of my white blood cells bumps into it, they're like, something's wrong, kill it. So my cells are supposed to tell on themselves. It's almost like pretend that I have some disease and 10 minutes from now, I'm going to explode and take all of you guys with me. It would be better if I told on myself and I could be removed before I like destroy everybody around me too. Yeah. It can. So if a cell knows that something is wrong, it's supposed to do this. There are other things that it does too that we eventually talk about, but this is one of the things that it can do is it can flip those phospholipids in and out. So this is just an interesting reason for like, hey, why is the outer layer and the inner layer different? Well, one reason is it's a way that I can tell everybody like, hey, I'm sick, you should probably destroy me. And if I have one liver cell that dies, we'll just grow another liver cell. Who cares? If I have one skin cell that dies, I'll just grow another skin cell. Who cares? So bizarre. All right. Other questions so far? If you guys ever have questions, just stop and ask. All right. So I got these phospholipids. One last thing we'll mention about them before we start talking about proteins is I can also add cholesterol in there. So I don't know how well you can tell from there. I'll zoom in a little bit. But cholesterol has this distinctive hexagon, 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 pentagon shape. So that's what that's supposed to be, hex, 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 pentagon. And if I put cholesterol in there, it can change the fluidity a little bit. For AP Bio, all you have to know is it changes fluidity. It's kind of weird when the temperature goes up and they'd be getting more fluid. If I add cholesterol, it kind of solidifies them. When the temperature's going down and they would be getting more solid, adding cholesterol makes them more fluid. So as long as you just knew that like, hey, I can tweak how much cholesterol's in there, and that can help determine whether this cell membrane is more liquid or more solid, you're good to go. So here's cholesterol again. Hex, 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 pentagon in like a stair-step kind of fashion, like that. All right, cool. So the next thing that they did was this. Somebody was looking at some cells one day, and they're like, all right, I got these phospholipids. I want to see if I can like see if there's something in the middle of them. So now, imagine that you had the task of trying to cut between the phospholipid bilayers to see what was inside. Or I just wanted to peel off the purple layer to see what was in the middle. Again, it's kind of like my peanut butter and jelly sandwich. Pretend that I tasked you with this is a phospholipid bilayer. I want you to peel off the bread to see the peanut butter in the middle. So I want you to somehow peel apart the phospholipid bilayer. Talk with somebody around you. How in the heck could you do that? I want to peel it apart like that. It's 1/9,000th the thickness of a sheet of paper, and this is like 70 years ago. All right. Very difficult question. Anybody got an idea? Look at that. Just friends calling you out. I can't just sort of like take a scalpel, even if I could get like a really, really, really fine scalpel. That's how, like, if they have an individual cell and they're wanting to like suck out the nucleus and input another one, they just under a microscope by hand do that. Like in vitro fertilization and stuff like that, they just like by hand do that. So, but even if I had a really, really sharp scalpel, if this is like phospholipid bilayer and I try to cut through it, it's oil, so won't it just like go right back together? Kind of like if I have like a, you know, I pour some olive oil up here and then I try to cut it, like, well, that didn't work. So what they did was they froze it. So I've got this phospholipid bilayer, and they freeze it, and then they just like shake it really hard. And when they do, parts of it rip off. So it's the most non-scientific way you could possibly separate stuff. If you have trouble cutting things, then just freeze it and like shake it really hard, and it probably falls apart. So I've got this phospholipid bilayer like this, and when they cut it, all of a sudden they saw this crap sticking out. Looked like a mountain range. And so they said it kind of looked like a fluid mosaic, so they call the cell membrane, your book might refer to it as a fluid mosaic. But the chunks sticking out were like all of these proteins. So imagine I removed this top layer of phospholipids. Could you see how these purple proteins would all be like sticking up, like half of them would just be sticking up kind of like mountaintops? And so that's what they saw. And so they're like, all right, that's probably important. And it was. So now what we're going to do is look at cell membranes, and we're going to kind of talk about all different types of membrane transport all on one picture. So if you're the type of student that like writes down stuff whenever I write it on the board, or if I draw a picture on the board, you draw it, then the way that this would work is for like all of this class and like a pretty good chunk of next class, there's just this one giant picture that I make. So if you kind of go along with me, then I would like take an entire page and just make a giant oval. What I would do if I were you is, give me 1 second, I'll kind of show you. My picture won't look exactly like this, but what I'm trying to show you is make sure you leave space on the outside because you end up writing some stuff on the outside. So I'll show you what my picture is going to look like. Of course, these are your notes, so your picture doesn't have to be identical to mine. But I kind of draw a little bit of phospholipid bilayer in one corner. Let me turn down my volumes there. And then after that, I just kind of like continue it like that. So this is sort of how I do mine. And what this does is we put most everything from the rest of chapter 5 like on this picture. So it's one big picture. And the reason it's good, and I got to work on drawing my ovals there. That was great for like 2/3 of it. Now I don't know what happened. Anyway, so we'll put most stuff dealing with phospholipid bilayers like all on one picture. And that also lets you know that in one cell, all this crap is happening at the exact same time. It's actually pretty useful. All right, so first off, what's it called when stuff moves from high concentration to low concentration? Diffusion. There's a couple types of diffusion. So what you guys learned as diffusion back in 9th grade or chemistry or whenever is actually simple diffusion. We're going to learn a second type of diffusion here in a little bit. So that just says simple diff, if you're having trouble reading it from where you're at. So simple diffusion. All right. So with simple diffusion, certain types of things literally just slide between those phospholipids. So since the phospholipids are always moving, back to this, here's my phospholipid bilayer. They're always just shuffling around. Hang on. There we go. Then from the side, aren't there occasionally like little gaps where like I could maybe see y'all through like one of these gaps? So if something is small enough, it might get through. Like literally I could have like some of these dots right here. Some of these molecules, whatever they are, they might be able to slide through from high to low concentration if they're small enough. Right, that makes sense. If they're too big, they can't really squeeze through the gaps very easily. So smaller things get through the gaps easier. That makes sense. At your home, if you got a window and you want to open it up, but you don't want to let in like mosquitoes or whatever, there might be some kind of like mesh on there, a window screen, and now like air gets through easily, but mosquitoes or bees or something, not so much. So for simple diffusion to work, one thing is size. It's got to be small enough. The second thing, though, talk with somebody around you. Which things do you think slide through more easily, polar things or non-polar things? Polar or non? What gets through easier? Polar or non-polar? All right. So we'll vote. Assuming that the particles are both super small, equal size, raise your hand if you think the polar things get through more easily. Okay. Raise your hand for non-polar. It's about 50-50. So it turns out non-polar things get through more easily, polar things not so much. So let's say that these particles right here are small and non-polar. They can slide through. That just says small plus non-polar. These orange ones over here, maybe they're like large and polar. The larger you are, the more likely you are to have difficulty getting through. And the more polar you are, the more difficulty you'll have getting through. So small and non-polar things slide through more easily. Large polar things, they kind of bounce off the membrane a little bit. All right. This has puzzled me for my entire career. So if you go off and you're in some cell biology class, I've been throwing this out there for like 2 decades. Nobody's gotten back to me yet. So when you guys go off to college, if some professor explains why this works, I'd like to know. So I can explain why large polar things can't get through. I can 100% explain that. I can only 50% explain this one. Okay, so large polar things. What's the environment like around the cell? Like what's the main thing that the cell is like floating in, so to speak, to give you a hint? Yeah, water. So something that's polar, isn't it like perfectly stable, hanging out in the water? Yeah. And then inside the cell is watery also. So it's a water-based environment in there. It's not pure water, of course, but it's a watery-based environment. So here's what makes sense to me. This particle is perfectly stable out here in the water. I understand that it's perfectly stable if it gets close to those phosphate heads. So maybe it even makes it in this far. But I also understand that once it starts hitting those lipid tails, that's kind of a barrier. So I understand why the polar things can't get through. They're perfectly stable out here in the water. They're perfectly stable right next to the phosphate heads, but they don't make it past that lipid barrier. Just like when water's trying to escape our lips and we don't want to have chapped lips, we put on chapstick. The water can't get past that waxy layer, so the moisture stays in my lips. As long as I put a layer of something non-polar on there, like chapstick, then I don't get chapped lips. If I put water on there, that doesn't help It just evaporates. So that totally makes sense why polar things don't make it through. So I've read I don't even know how many cell biology books and sources on this. Here's what they all say. This nonpolar particle, is it very stable out here in the water? No. Is it very stable next to the phosphate heads? No. Is it stable next to the lipid tails? Yes. So I understand why this nonpolar particle would make it in to the layer where the tails are. What I don't understand is why it keeps going. But it does. And I don't understand. That's why that's a frowny face. I don't get it. That was just coincidence, but I'll go ahead and... So I don't understand why it keeps going. In all books, they'll just be like, and then it keeps on going, and you're like, but why? And they're like, next. So nobody explains why it keeps going. So if you are at the University of such and such in the future, and somebody tells you why it keeps going, I'd like to know. I'm assuming some protein pulls it through, but then it's less likely to be simple diffusion. And again, books never say that. So one of the great mysteries. Like, what'd you do this weekend? I thought about that. That's what I did. Not really. Okay, so simple diffusion is when things move from high concentration to low concentration. The smaller you are and the more nonpolar you are, the more likely you are to get through. The larger and more polar you are, the less likely you are to get through. All right, cool. What if I have something that I want to get through that's polar or pretty big? How do I do that? Like, let's say that I want these orange dots. How can I get those in there? Yep, some kind of protein. So I got different versions of proteins. Which things might allow me to let something pass on through? Yeah, channel. That's just sort of like a little hallway or a little doorway. So what I can do is I can have, number 2, facilitated diffusion. Facilitated. And that's probably spelled out somewhere in your notes. I always forget if it has 1 L or 2. Based on my spacing, it's only going to have 1. But if your notes say that it's got 2, then I guess it's got 2. So facilitated. What does it mean if I facilitate something? Yeah, I allow it to happen, or I encourage it, or help it to happen. As a teacher, I should facilitate learning. It should be easier for you to understand this stuff if I'm here, and harder for you to understand stuff if I'm gone. So for some teachers, that might be true. I had teachers that wasn't always true. It was sort of like, I'm going to learn about the same whether you're here or not, in some classes that I had. But facilitated means help. So it's help diffusion. Thank you. Yeah. Oh, you good? Yes, I have seen all of them. Thank you. You too. All right, so facilitated diffusion is helped diffusion. Over here, nothing really helped me. They were just kind of on their own. But I could put in a channel, and I just sort of make like a corridor that would let something in. So one of the ways that I could do facilitated diffusion is with a channel. And they use the word channel kind of like you would in like a geography class or something. You know, if I have like a body of water and there's like a little connector, they call that a channel, or a canal, like the Panama Canal. So it's kind of like that. So one type of facilitated diffusion is a channel. Okay, this is typically how we draw channels. It'll usually just have like a little oval like there and there, or it might just be like a solid block, like 2 rectangles. That's how they usually draw channels. One of the things that it doesn't show, though, is channels are usually highly specific. So it looks like anything that's kind of small could slide through there, but that's not the case. it turns out that I have different channels to let in different individual things. So I have a channel called aquaporin, and it only lets in aqua. It only lets in water. I have sodium channels. They're really good at only letting sodium in. I have potassium channels. They're really good at only letting potassium in. So even though this looks like just a wide open hallway that anybody could walk down, it's not. So it's not like, pretend that this door is a channel. This room is a cell. It's not quite like the door to where just anybody less than like 5,000 pounds could fit through the door or something like that. You know, this guy, he's like, he can't. He didn't walk up here anyway. But so the 5,000-pound man can't get through the door. But what it really is pretend it's like a perfect cutout of just you. And you're the only person with your exact proportions. So it really only lets you in. And then there's a different channel that lets like this other student in. And there's a different one that's got a perfect cutout of this third student, and it only lets them in. So most channels are highly specific, and they rarely let in the incorrect thing. It might be 10 million to 1, a billion to 1. 1 out of a billion times they accidentally let in the wrong thing. So they're actually highly specific. So that's the only thing that's kind of lacking in the drawing. They're highly specific. For a long time, when I was in AP Bio, I graduated high school in '94. They finished the Human Genome Project like 10 years later. So when I was in high school, we didn't know anything. Like there was so much stuff we didn't know. And so when I was in high school, I was taught water just somehow slides through, and we don't know why. When they started checking water, and they're looking at how fast water gets through, they're like, hey, water's getting through the cell like, I don't know, about 1 billion% more than we predicted. It wasn't like 5% more. It was billions of percents more. So they're like, all right, small things get through, non-polar things get through, except water. For some reason, like water just slides through the membranes. We don't know why. And then they discovered this channel that was made just for water. And that's how water gets in. So I have channels just for certain things. All right. The problem with this is now, anytime I've got a bunch of these orange dots on the outside, they're going to always go from high to low inside. They're going to always go from high to low until they reach equilibrium. So things will always be moving. What if I don't want them to move right now? Then what could I do? Yeah, I put like a little gate on it. So now I can like open and close the channel whenever I want, just like this door. If I have it like this, then people can roam in and out free will, even if it's specific. But if I close the door, now they only come in whenever I let them in. Turns out this is how you have conscious thought. When I choose that I want to think about 4 plus 4 is 8, as opposed to something stupid like 3 plus 3 is 6, I think we can all agree on that. Then I can selectively like open and close these doors. We learned later on in the nervous system that letting sodium and potassium ions in and out of cells is what a thought is. A T-H-O-U-G-H-T thought, just to be clear. So I can choose when to think about something or not think about something by like opening and closing these doors. So number 3, I can have a gated channel. And it works exactly like you would think, a gate. I've got a channel, but I've got a gate that I can open and close. So whenever I open it, these large polar things could go through, and whenever I close it, even if they want to move from high to low, they can't. A gated channel. A gated channel. So I can open and close that. Just like this door, it's homecoming week. So pretend this was a perfectly sealed door. There's no cracks around it. And they released like purple smoke in the hallways. They're like, homecoming, Timber Creek. And then they're like, they're going to choke and die. Nobody thought about that. But like purple smoke in the hallway, it doesn't enter the room. But if I open up the door, then it will move from high to low, right? So it's just like that. I can decide when to let something in and when to let something out. So gated channels are how I can actually have like my own thoughts, which is super bizarre. So weird. Real quick, if you're like, all right, up here, all these are just blobs. I know they're proteins, so what are the building blocks of proteins? Yeah, amino acids, chains of amino acids in the right order, and then they fold in all kinds of shapes. These are some various channels. Got a bunch of alpha helices. This is what they call a beta barrel, because it's made out of beta pleated sheets. And if you look at it from the side, it looks like that. But if you look at it from the top, it looks like that. You're like looking down the barrel. You're looking down a beta barrel. And so that's a channel, and something can like go through there. All right, we could probably use something interesting. This is a protein called hemolysin. You don't have to write this down. Hemolysin. This is basically a word with 3 parts. What's that IN tell me? It's a protein. What about hemo? What do you think of when you think of hemo? Blood, yeah, like hematocrit, hemoglobin, stuff like that. And then what about lyse, L-Y-S? L-I-C-E is like hair lice, but what about like L-Y-S? Like lysosomes, Lysol. What does lyse mean? It means to rip apart. So lysosomes, we'll learn, those are we always call them like the custodians of the cell. They would like break down trash. So they chemically destroy stuff. So this is a protein that destroys blood cells? Yeah. So it turns out there is this type of staph infection that I can get, and the bacteria manufactures hemolysin. It then shoves this into my cells, and that's a beta barrel. So basically pokes holes into my red blood cells, and then they explode. And that's why I get sick if I have a staph infection. So it literally just takes these things, erythrocytes are red blood cells, and it's just sort of like this bacteria builds these proteins and then just like inserts these random channels into my red blood cells. Our red blood cells explode. They release iron, and it turns out that that's what was keeping the bacteria from growing really well was iron. So now that there's iron everywhere, now they grow like crazy. It's almost like I went into, like we're starving, and we find like the food supply, and we blow open the walls. Now that there's food, we all start like reproducing like crazy. So who would have thought? Channels can also be used against us. Super weird. No. It's like, oh, that protein looks so cool. And you're like, yeah, until it kills you. But before that, it does look pretty cool. All right. So I've got channels. We do have one word for, let's say that this happened to be water. We'll do number 4 here. What if I had a channel and it happens to be water that's moving from high to low concentration? What was the name of that one? This is a word that everybody's heard. Osmosis. So for some reason, if water's moving through a channel, they call it osmosis. I don't know why they don't just call it water diffusion. But they call it osmosis. So I've got simple diffusion, bless you. Small things, non-polar things can literally just squeeze between phospholipids. Facilitated diffusion, I put in a channel, and now if something's too large or too polar or both, then I can make a channel specific for it and let it move in and out at free will. If the stuff that's moving in and out at free will is water, we call it osmosis. And then if I want to control when stuff can get in and when it can't, then I can put a gate on there, and that's a gated channel. All right, cool. So now, so far, everything that we have up here at this point is passive transport. everything up until now is passive transport. Everything after this is going to be active transport. So however you do that, if you want to like put an arrow, but like 1, 2, 3, 4, those are all passive transport. Number 5, the next one we're going to look at is active transport. So 1, 2, 3, 4 is all passive transport of stuff. Some sort of molecule, sodium, potassium, water, whatever. That's all passive. After that, it's active. So which one would require energy, passive or active? Active. And then cells run almost exclusively. What was that 3-letter molecule that cells run almost exclusively on? ATP. So we'll learn a little bit later. There's some exceptions, but ATP is what most cells run on. So it's kind of like everything in this building runs on electricity. You know, this and this and this and the wireless, the AC that's on, pretty much everything in this building runs on electricity. I don't know of anything like in the building that actually runs on like gasoline. Maybe we've got some hot water tanks that run on natural gas, but everything else runs on pretty much electricity. Same thing with our cells. Our cells run almost exclusively on ATP. So if it's active transport, I always need energy, and it's usually ATP. I will always need energy, and it's usually, that says usually, let me try that again. Always need, capital E is energy, and then that energy is usually ATP. So there's ATP, CTP, GTP, UTP, all 4 letters in RNA. A's, U's, G's, and C's. There's a ATP, a GTP, a CTP, and a UTP. Out in nature, we use this one almost exclusively. Just like electricity, we use almost exclusively, but some stuff runs on gasoline. Some stuff runs on kerosene. Some things run on something else, you know, natural gas. But usually ATP. So the most common active transport I draw as like a rectangle is called a pump. So it's a protein pump. All of these channels and stuff are all made of proteins. It's a protein pump. All right, so active transport requires energy, probably ATP. The first example is a pump. Anybody tell me why I would use a pump that uses energy when I could just use a channel that doesn't? Yeah. Yeah, to basically hoard, with a D, to hoard particles that I want inside without them just like leaving. If I have a channel, then I can't stockpile a bunch of large polar molecules. If there's 100 of them out here and 0, then like they'll move in until they're 50-50, and that's all I can store. But what if like there's 100 large polar molecules and I want all 100 in here, I want to stockpile them. I'm going to need a pump. So normally things move from high concentration to low concentration, but with pumps, I can like stockpile stuff. So now, let's say that there's a bunch of these purple dots. Yeah, we'll do it that way. And pretend I don't like purple dots. So even though there's more outside than inside, I want to get rid of all of them. So I can pump them from low to high concentration. If there were a bunch inside and I wanted to get more inside, I can pump them inside from low to high. So with a pump, I'm normally going from low to high concentration. And that requires energy. Things naturally move from high to low. But with energy, I can make them move from low to high. Naturally, water flows downhill. But with energy, I can get a pump, and I could, like, put it back up at the top of the hill again. So these large polar molecules here will move in until it's about 50-50, and I have no real control. But if I wanted to get rid of all these purple dots, I can just pump them out. So we have like sodium pumps and potassium pumps and chlorine pumps, all these different pumps to kind of control what gets in and what gets out of my cells. Right now, this next one, number 6, they call it co-transports and counter-transports. Let me write that differently. That makes it look like they're all one thing. So I have co-transports and counter-transports. Co-transport and counter-transport. Co-transport and counter-transport. So this one is less likely to be on the AP Bio test. It'll show up in diagrams, but as far as you having to explain it, it's less likely. Explaining everything before this is totally fair game, shows up a lot. It's almost like factoring in an algebra class or finding a derivative in a calculus class. Co-transport and counter-transport would be more like if they wanted to ask you, they're like, hey, we need a hard cell membrane question. Okay, co-transport, counter-transport. Okay, so co-transport and counter-transport. They classify it as active. That's why it's on this side of the line. I don't think they should, but they do. So here's how it works. I've got this big old protein right here, and pretend that I've got a lot of purple dots inside the cell and not very many out. All right, cool. And then pretend that I've got a lot of blue particles on the outside, but not very many on the inside. So if I'm a student and I don't have like different colored pencils or pens, then like, all right, the purples are dots and the things in bluish green are like triangles or something, or pluses, or maybe not plus, that might confuse you, but like you can use different shapes if you don't have different colors. All right, cool. So here's kind of how this works. Pretend that I don't give a crap about purple dots. So which way will the purple dots naturally move? If I don't have any energy, pretend it was passive transport, would they be moving into or out of the cell? Out. And we know that because they'd be moving from high to low. So I just let them move from high to low. However, let's say that I hate these blue dots. If I want to get rid of them, which way do I want them to move? In or out? Out. But naturally, will they move out? No. So I somehow have to move them out without burning any ATP. I would call this passive transport. Textbooks call it active. I think it should be called passive because the cell doesn't burn any energy. Take maybe 20 seconds or so. How is it that I could move these from low to high without using any of my own energy? It is related to the purple guys. All right. Anybody want to take a crack at it on their own? if things were going to go from low concentration to high, that would require ATP or require energy from the cell. The cell would be spending energy. But in this scenario, the cell is not spending energy. So where's the energy coming from to push those out? From these guys moving. It's kind of like these guys are pushing the gears. I don't care about the purple dots, so if they leave, fine. That sort of like turns the crank to push those guys out. Let me give you an example. If you ever go to like an airport or a fancy hotel, that's how you know your hotel is fancy, if they have one of those revolving doors. Or if you go to an airport, a lot of times they have revolving doors. Back in the day, you had to push them. This is modern America, so you just walk up and it starts spinning for you, right? Have you guys ever seen the ones that you actually have to push? If not, you can imagine it. Back before everything was motorized, you know, they didn't have, it's a tough world out there. Like if you're in a wheelchair, you just went up the stairs backwards. And you just got good at going downstairs. So I don't know when they started like making everything like wheelchair accessible and everything. But like doors, they weren't motorized. That's within like the last 20 years. We go up and like push the thing and the door opens. The only motorized doors I grew up with were like these at the grocery store. So it was different. So anyway, let's say that you go to an airport. And let's say that there's like a rotating door right here. And so you know how from above it would look like sliced pizza or like sliced pie? Like there's sort of this little like triangle and I'm in this part and somebody else could be in this other part, somebody else could be in this other part. Is this making any sense at all? Okay. So now pretend I want these to like exit the cell. So we're inside this room. This room is the cell. And here's this blue dot. I want it to leave, but it's going to require energy to make it leave. So pretend that it's like loaded into this rotating door. So it's right here. It's kind of like a baby, like a 2-year-old baby that's like too weak, maybe a 1-year-old. They're like stomping around. It's a 1-year-old and it's a girl. She's walking. If it's a boy, he's still crawling because he's an idiot. If you've ever heard that girls mature faster, they mature faster. If a girl isn't walking at 1 year old, we're concerned. If a boy isn't walking at 1 year old, we're like, yeah. So you got the 1-year-old baby. They're kind of in there. They're not strong enough to make it go. So they're like loaded in there. So the whole thing isn't moving. Then I come along. I have the power to make it move. So as I push it, that just sort of like pushes the baby on out, right? So he went from low to high concentration because I went from high to low. I provided the energy, the door did not. I did the pushing, the door didn't. And now the baby's outside. So the power of me passively moving is used for the baby to like hitch a ride. Let me go get my baby. All right. Nah, it's just parenting. All right. Same idea, except now I want the baby inside the room. So baby is loaded up in there. Baby's not strong enough to do any pushing. It's going to require energy to move from low to high. And so now the force of me exiting the room, as I like push on this little spinning door this way, then now the baby goes into the room more gently as a better parent. So now the baby goes in that way. Do you see how the door didn't provide the energy? It was the passive transport of the purple dots was providing the energy to like move this guy across? Yeah, so they call it co-transport or counter-transport. What do you think the difference is between co versus counter? I showed you both. That might be true. What's an easier, I'm not saying you're wrong. What's an easier way of saying it? Pretend that I couldn't even see how many dots of each I had. If one's going one way and one's going the other way, it's counter. If they're both going together, cooperate. If they're both going the same way, they call it co-transport. So the second example, where I have an arrow going out and an arrow going in, that's counter. Co means together. Again, cooperate. Counter is like the opposite. So somebody attacks, and so I counterattack. We're having some sort of debate, like a speech. Somebody has a point, and so I make a counterpoint. It's like, well, no, that's not going to raise taxes. That'll actually lower taxes. And so I kind of, counter means opposite. So basically, it's all which way are the arrows going. If the arrows are both going out or both going in, that's co. If one's going one way and the other's going the opposite way, that's countertransport. Again, this is classified as active transport. I think that's a bad name because the cell doesn't have to burn energy. If it's done perfectly, the cell doesn't have to burn energy. So to me, I think it should be called passive, but all textbooks classify that as active transport. So I guess you can think of, yeah, there's energy, but this guy's providing it or something. I don't know. All right, now, these are actually the least interesting of the proteins. So now that we're moving on to the most interesting ones, I'll tell you that those were the least interesting ones. So now, let's look at another type of protein. This guy right here, a glycoprotein. All of these guys were actually moving stuff in and out, like physical things like these particles are moving in or these particles are moving out. The next one, we're just sort of like conveying information, but nothing's moving in or out. So number 7 will be a glycoprotein. 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 All right, protein means protein. Does anybody know what glyc or glyco means? Yeah, glucose, carbohydrates. You might have heard of glycogen. I store glucose as glycogen in my liver. If you ever exercise like super, super hard, like sprint level hard, or like CrossFit where they're like, do as many push-ups as you can, and then like go run down there and back as fast as you can, and then do as many push-ups as you can, and after like 8 minutes, you feel nauseated. That's because your blood sugar is really low. So if you ever like exercise really, really hard, and inside of like 10 minutes you feel nauseated, that's low blood sugar. And what happened is you used up all of the sugar that you have, you dumped all the glycogen from your liver into your bloodstream, and then you kept on running, and so you feel nauseated when you have really, really low blood sugar. All right, so glyco means sugar. You might have heard of the process glycolysis, glycolysis. That's splitting, lysis, sugar. So glycoproteins, it's a protein, and then these are hexagons. I don't know quite how well you can see it from where you're at. Let me, those watching the video, do that. But if I look at these kind of zoomed in, you might be able to tell that they're like hexagons. I can't see it, but I know they're there. There's hexagons with oxygens in the corner. So those are carbohydrate chains. So carbohydrate protein. The protein is there just kind of as an anchor, so to speak. And glycoproteins, their main function is cell identification. Cell ID. Cell identification. Glycoproteins are for cell identification. It's not the only thing they do, but it's one of the main things they do. There's another cell membrane. There's a protein. Hexagons coming off of it. They should have an oxygen in the corner if we were zoomed in enough, so that's a glycoprotein. Over here, if I have sugars attached to a lipid, they call it a glycolipid. They both have the same idea. Glycoproteins and glycolipids are there for cell identification. So earlier, I mentioned that like if I'm a white blood cell, I go around and I check this one, and I'm like, hey, this is a Mr. Cotton lung cell. How do I know it's a Mr. Cotton lung cell? It has cell ID on it that says Mr. Cotton lung cell. This is a Mr. Cotton liver cell. How do I know it's a Mr. Cotton liver cell? It has different glycoproteins on it that identify it as a Mr. Cotton liver cell. How many of you all know your blood type? Quite a few. Okay. So your blood type, your A, B, AB, or O, and then you might have this RH factor. If you have it, we say you're plus, and if you don't, you're minus. So I might be like O positive or O negative or something like that. Well, basically what they did was on your red blood cells specifically. Do I have a red? Okay, I guess I don't. So red blood cells are shaped kind of like that. They're sort of like concave in the middle. Sort of like that. And there are proteins on there. And if you have like these proteins that have a certain shape, we called it A. And you have blood type A. If instead your red blood cells have this other shaped protein on them, maybe like that, we name that protein B, and you have blood type B. What if I'm blood type AB? I got a little bit of both. So I have some of the mountaintops and then some of the like little half circles. I'm blood type AB. If I'm blood type O, I don't have either one of them. So what happens if I have blood type A, I'm dying on the side of the road, you pull up, you have blood type B, so you take out your plastic tubing and your little needle, and you're like, well, I'll just, transfuse this guy like on the spot. What happens if I have blood type B and you have blood type A and you transfuse me with blood? Yeah, it turns out it turns into a solid. When my immune system attacks the blood that doesn't correspond to mine, it sticks and it clots. So it actually, the reason I die is because my blood just clots. If you've ever done your blood type, again, everything was different back in the day. So when I was in college, in anatomy lab, or physiology lab, in anatomy lab we dissected. In physiology lab, we were always either pissing in a cup, and then we analyzed it, or we pricked our fingers to get blood and analyzed that. I bet 85% of the time I was in lab, you'd walk in, and they either have like all these glass slides and a bunch of needles, and you're just like, so that bruises from last Thursday. So you go over there, and they're like, all right, we're going to type our blood. So if you ever, again, this is back in the day, and like AIDS had been around for like 13 years, and we were still doing this. They're just like, be careful. So what you do is you get like a glass slide, like a microscope slide, stab your finger, drop of blood, drop of blood. So there you go. I got 2 drops. Then we had anti-A serum and anti-B serum. You might have heard of antibodies, anti-A antibodies, anti-B antibodies. So I basically take this bottle of anti-A and I put a drop of it on the first drop. And I just look at it. No microscope, nothing, just naked eye. Nothing happens. I'm not A. Then I take some anti-B and I put it on the second drop. And I look at it. And before my eyes, it turns into a clump. Not on a microscopic level, like it looks like cherry slushy. So now imagine that I get transfused with like a whole bag of A and my body attacks it. And that whole bag turns into like slush. I have a heart attack and die. So 150 years ago, if somebody was bleeding to death, they would transfuse them, and they're just like, sometimes it works and sometimes it kills you. We don't know why. And they would just do it. Then eventually they found out, like, hey, if we transfuse you with like a close relative, you're less likely to die. And then eventually they found out that's because if I'm type A, one of my relatives is more likely type A than the general population. So anyway, these are glycoproteins that identify you as like, I'm type A blood, or I'm type B blood, or it's like, I've got both. And then your immune system either attacks or doesn't attack. So if I'm type A and you give me type A, my immune system thinks it's my own blood. If I'm type A and you give me some type O, neither of those proteins are on there, so it's cool. There's nothing to piss off my immune system. My immune system, if I'm blood type B, I hate these triangles. Well, that doesn't have the triangles, so we're cool. Did you have your hand up?
Yeah. So are those tests actually accurate? Like,
Yeah. Now, when you say those, like the one I did was accurate. If you're talking about like a different test like that you can buy or whatever, it's got to be. The only way that they would be inaccurate is if like the antibody was old or something like that. So as long as everything was fresh, like we just had bottles of anti-A, and it really was just like, hey, wash your hands, here's a glass slide, drop of blood, put a drop. Like it wasn't a whole lot of measure this, look at that. Like I could be this far away from the slide and it turns chunky enough that from like 3 feet away, I'm like, that one's turning chunky. It actually looked like cherry slushy from Quick Trip or something. So super bizarre. Glycoproteins. All right. Another thing that's interesting. We do talk about cancer a lot. I know that hits close to home for probably everybody. Most everybody knows somebody, a loved one that has passed from cancer or is currently fighting cancer, etc. But this relates to cancer also. So my mother-in-law, my wife's mom, she was at the time like early 50s, like 52 or something. I'm 49, so she was like just a few years older than me. She's 52, and she was just out like riding lawnmower, mowing the grass. So she's coming up, there's a tree with kind of a low branch, so as she's doing the riding lawnmower, she just kind of like ducks like that, breaks her rib. And it's like, you know, she's not like into fitness, but like when you're 52, that shouldn't break a rib. And so they go and they check, and they're like, oh my gosh, like it looks like there's something wrong with your ribs. They do all kinds of scans and x-rays, and they determine that there's cancer in the rib. So then you're like, okay, well, was it bone cancer? No, it was lung cancer. How do they know that it's lung cancer? They can do what's called a biopsy. And let's say that I've got a lump. Pretend I have a lump. Cancers always cause tumors because cancer is cells growing out of control. So you'll always just have this extra lump of cells. So pretend I got this lump of cells like an inch into my abdomen. So it's under there. What they'll do is to get a sample of it, they have a needle, picture just like a syringe, kind of like you're going to get a shot or something, that kind of needle. And they have a little cover on it, and they can like poke through the skin, poke through the muscle, poke through whatever. Once they're in there, they do some little thing right here, and it removes the cover, and now they just kind of like stab the cancer. They stab the tumor. And then when they take it out, it's got like a little bit of the tumor like stuck in the needle. Right? Imagine if there was like an apple right there, and I have a metal needle, and I stab it. Now don't I have like a cylinder of apple stuck in my needle? That's what they do. How do they not get skin and muscle and everything else? They have the little cover on there. So at first they're piercing my skin. Then they're able to like remove the cover somehow. And then they like stab the cancer. So that's what a biopsy is. So they go in and they get like a small chunk of this tumor, and then they can look at it under a microscope slide. And even though this tumor maybe came out of my rib over here, they can check the glycoproteins on it, and the glycoproteins identify this as a lung cell. So then they're like, oh, crap. Well, now we need to do a scan of her lungs. And then you look at her lungs, and they're full of tumors. That's how we know that the tumor started in the lungs, because it's lung cells in my bones. And I know they're lung cells because the cell ID tells me so. Just like this says, this is a red blood cell type A. These proteins mean this is a red blood cell type B. That's how we know. So if you've ever known somebody and they're like, hey, they discovered cancer in this location, but it turns out it was actually liver cancer that had moved to the lungs. It wasn't lung cancer that moved to the liver. It's like, how did they know? I can check the glycoproteins on there. So usually if cancer stays in one spot, we've got a decent chance of treating it. When it starts moving around, that's when it gets tough because you can't cut it out. So if that cancer all stays in one lump right here, I can cut it. But if it's moved to other places, I can't cut out everything. And then how do I know it hasn't dropped off cells along the way? So that's when it gets tricky. So we always try to catch them before they move. So cell ID, we're done for today. See you guys later.